continue with this nuclear endosperm. Now in this picture you can see only 4 diagrams, that is A, B, C and D, where the central vacuole has enlarged and it has pushed all the nuclei towards the periphery. Now I will take you to another picture which has a complete journey. This is a diagrammatic representation of free nuclear endosperm and its cellularization. The first few we have already discussed, where we have the primary endosperm nucleus, then there is division in this primary endosperm nucleus. The third one is the cenocytic or the syncytial stage, where there are multiple nuclei which are present in the cell and there is no cell wall formation. Now with the enlargement of the central vacuole, there is migration of nuclei towards the periphery. Eventually, the cellularization starts happening in a centripetal manner, and this cellularization starts from the margin and it moves towards the center. The vacuole which is occupying the central position, it gradually disappears and the cell divisions, they continue towards the center, and you can see that the last stage there is total cellularization of the endosperm. So if I have to give you few key points about this type of endosperm is that there are free nuclear divisions which happen, and the major portion or the major journey of the development of the endosperm is by free nuclear divisions only. It is only towards the maturity that the cellularization occurs, and that too occurs initially at the periphery and it moves to the center. Coconut is a good example. Initially, the primary endosperm nucleus undergoes a number of free nuclear divisions. Now when the fruit is about 50 millimeter in length, the embryo sac remains filled with a watery fluid or the coconut milk, and this contains free nuclei and also fine cytoplasmic particles. Later, when the fruit is about 100 millimeter, the liquid in addition to these free nuclei also shows several cells, each enclosing variable number of nuclei. I am sure all of you would have seen a green coconut. So when you cut open a green coconut, there is lot of water which is present, which we say is the coconut water, okay, which is very healthy, and that is what is the watery fluid which I am talking about. And if you break open a ripe coconut, there is very little or no water which is present, but there is white meat which is present in that coconut. Because the free cells, they gradually settle on the periphery and they form a white gelatinous layer. Subsequent divisions increase the thickness of this gelatinous mass. This gelatinous mass is also the cellularized endosperm, which is commonly called as the coconut meat. The coconut water in a mature coconut is devoid of any nuclei and cells. One other example is Erica catechu, which also shows a similar sort of endosperm development. Here you can see, the first one you are familiar with. The second diagram which you see is, on maturity of coconuts, the endosperm does not have free nuclei or the free cells, because there is cellularization which takes place. Another example, this is free nuclear endosperm development in Arachis hypogaea. Do you know what Arachis hypogaea is? In A, the zygote is at the top and the primary endosperm nucleus is in the center. First division in the primary endosperm nucleus gives rise to a 2-nucleate primary endosperm cell, right? So after that in C you see there is a 4 nucleate primary endosperm cell with starch. Pro-embryo is present at the top. And then in figure D you see that there is a cenocyte with globular pro-embryo which is shown here as EM. Then there are endosperm nuclei seen dividing, and in free nuclear stage these are present around the globular embryo. In certain species, the nuclear endosperm is also accompanied by Haustorium formation, and this Haustorium develops from the acellularized portion, that is that portion which is not cellularized. It is commonly seen in Cucurbitaceae. The structure of Haustorium, it varies among species based on cellularization. In some the cellularization is less, whereas in others cellularization is more. After the syncytium stage, only the micropylar region of the endosperm, it becomes cellular, while its callosal portion remains free nuclear, and this free nuclear portion, it will extend downwards in a tube like extension. So, this portion which is like a tube, it is the nuclear endosperm Haustoria which is present. The free nuclei of this endosperm, they migrate into this Haustorium, okay, or into this tube. Among cucurbids, Echinocystis lobata has the longest Haustorium which is about 16 millimeter in length. Cucumis species also has Haustorium, and here the tip of the Haustorium becomes spatulate. Do you know what is spatulate? Spatulate is spoon shaped. Now, in this particular picture, you see that there are 3 different species showing variation in the free nuclear endosperm Haustoria which are formed. In all 3 you can see that there are tube like structures towards the bottom, and these are the ones which are the Haustoria. And what is the function? It is basically for the uptake of nutrients from the adjoining cells. You would recall that haustorial cells, basically they are similar to transfer cells. Okay. So, here there are 3 species. The first one is Crotalaria, the second is Grevillea, the third one is Lomatia. In all 3 we see that there is a well developed free nuclear callosal Haustorium which is present. In all, the micropylar region is cellular. The extreme left, or the A, Crotalaria, it shows a spatulate end or the spoon shaped Haustorium which is present, right. Grevillea has a very cellularized micropylar portion and a small tube like portion or a Haustorium which is there, but this Haustorium does not show any branches. The third one, or the Lomatia, here you can see that there are many finger like projections which are present all along the wall of the Haustorium which is present. The second type of endosperm we are going to talk about is cellular endosperm. All nuclear divisions are accompanied by wall formation. It is a type of cellular development where karyokinesis is always followed by cytokinesis. So, whenever there is a nucleus formation, that is a nucleus divides, at the same time there is cell wall formation. The first wall is usually transverse but can also be vertical or oblique. The plane of division here is not constant. Mostly the endosperm, it becomes completely cellular, but there are exceptions like in Phaseolus, cellularization occurs only around the embryo. Now, here you see that this is a diagram depicting cellular endosperm in Drimys winteri. A figure shows embryo sac with zygote at the top and there is primary endosperm nucleus towards the middle portion of the cell or towards the center of the cell. Now, the first division in this primary endosperm nucleus, it is followed by cell wall formation, and it leads to two unequal sized chambers, the micropylar one and the callosal one, which you can see. Micropylar chamber is larger, the callosal chamber is smaller in size as compared to the micropylar one, and in figure D you can see that there is total cellularization of the primary endosperm cell. Here you see there is formation of Haustorium in cellular endosperm. I showed you some pictures where we saw that there are Haustoria developing in species where there is nuclear endosperm formation. Now, this Haustorium formation also takes place in cellular endosperm, but the formation of the Haustorium is variable. This one is endosperm in Barlaria, which is a member of Acanthaceae, showing both callosal and micropylar Haustoria. This is callosal Haustorium in Magnolia ovovata. You can see there is a thin tube-like structure arising from the cellular endosperm towards the bottom. Another example is Iodina rhombifolia. This is an endosperm with aggressive Haustorium. You can see there are many branches which are coming from the endosperm, right. Now, what are the key differences between nuclear and cellular endosperm? Obviously, the first and the foremost is that in nuclear endosperm, there are cell divisions and they are not followed by wall formation, whereas in cellular endosperm, whenever there is a nuclear division, it is followed by cell wall formation. That means there is no synocytic or syncytial stage which is present. There are no free nuclei which are present in the cellular endosperm. Okay. That is the major difference between the two, and of course, the process of cellularization does take place in nuclear endosperm, but then it takes place at a much later stage. Okay. Then we talk about the third or the Helobial type of endosperm. Now, this is called Helobial endosperm because it is a characteristic feature of the order Helobiales. It is defined by the features of both free nuclear and cellular endosperm, and this is the intermediate between the two. After the first division, one nucleus migrates towards the micropylar end and the other towards the callosal end. The divisions in the micropylar chamber are rapid and repeated, so they make it free nuclear. Later, these free nuclei, they will form walls and constitute a peripheral layer of cells, as we have already discussed in nuclear endosperm. After the completion of cell wall, centripetal growth occurs to form an enormous endosperm. So, what you see here is the nucleus of the callosal chamber does not divide, or if it does, it remains synocytic and finally degrades. Rarely this chamber becomes cellularized. This is an example of Helobial endosperm in Asphodylus. Another important point which I want to put forth here is the composite endosperm, which is seen in family Loranthaceae. Why is it called a composite endosperm? You can see that there are many endosperms which are seen here, and they all come together and fuse together to form a composite structure. In this fused mass of endosperm, there are several embryos which are present. You can see on the right hand side there are several embryos which you can see. Now, composite endosperm is a characteristic feature of Lorenzaceae. In the absence of true ovules, in Lorenzaceae there are no true ovules. So, all embryo sacs, they remain close together in an ovary. Soon after the primary endosperm nucleus is formed, it moves to the callosal part of a long and tubular embryo sac. Several divisions in the callosal portion of the embryo sac make it cellular. As all the embryo sacs, they lie close to each other, their separating walls, they dissolve. Thus, cellular endosperms from numerous embryo sacs, they come together and form a unique composite structure which is called as the composite endosperm. Then there is ruminate endosperm which you see here, which is in nutmeg. So, what is different here? You can see that there are some evaginations, some ruminations which are present on the structure. The endosperm does not have a smooth structure, it is uneven in surface, and this is seen in Myrstica fragrans. Ruminate endosperm is characterized by irregularities or unevenness. These are caused by either the seed coat activity or the endosperm itself. Any of the three types of endosperm may develop ruminations and become ruminate. Three types, I mean nuclear, cellular or helobial. Ruminations caused by seed coat may be due to an excessive ingrowth or invaginations in the seed coat, and they can also be due to unequal radial elongation of the cells of one or more layers of seed coat, as is seen in Passiflora myrstica. Myrstica of course is nutmeg. Now, what are the functions of endosperm? We have been talking about endosperm for quite some time. So, I would like to discuss the functions of endosperm. Endosperm, as already talked about, stores and provides nutrition to the embryo up to its maturity. The position of the endosperm between the embryo and maternal tissue clasa suggests its role in maternal nutrient transfer to the embryo. In cereals, the starchy endosperm, it contains food reserves which are used during seed germination and seedling establishment. See the function of the embryo. What does embryo? Embryo gives rise to a new plant, right? And this embryo, when it is developing, it is thriving on the nutrition which comes from the endosperm. In dicot seeds, the cellular endosperm is used as a nutrient source for embryo growth during seed development. During embryo formation, the endosperm also prevents the fusion of embryo with the surrounding tissues. That is, it ensures that there is a cover on the embryo and the embryo is not fusing with the cells which are present around it. Arabidopsis mutans, which lack endosperm cellularization, they produce very small and dry seeds because there is lack of nutrients during embryonic growth. So, this lack of nutrients leading to a very small dry seeds is because the endosperm, which is essential for the growth and development of the embryos and seeds, is not there. So, this confirms the role of endosperm in providing nutrition to embryo. The endosperm also senses environmental signals and then it secretes signals to regulate the growth of the embryo, because all times the environment cannot be favorable for the growth and development of the embryo. So, endosperm is the one which is guiding basically the embryo to go ahead further for germination or not. In the absence of endosperm, in families, like there are several families like Podostomaceae, Trapezae, Orchidaceae, where there is no endosperm which is present. Now when there is no endosperm, there are structures like Haustorium or Nucellar plasmodium, which are alternatives to endosperm, and they provide nourishment to the embryo. There are structures performing the function of nutritive tissue, and these can be the chalaziosperm or the perisperm. Here, there are some nutritive tissues other than endosperm. Of course, the primary tissue which provides nourishment to the embryo is the endosperm, but there are structures like pseudo-embryo sac we just talked about in Podostomaceae, pseudo-embryo sac or the Nucellar plasmodium. There is perisperm which is present in black pepper, and there is also chalaziosperm which also performs the function of providing nutrition to the growing embryo. In this figure, we see a fertilized ovule of a podostemad showing a pseudo-embryo sac. Now, pseudo-embryo sac is the one which is providing nourishment to the growing embryo. Pseudo-embryo sac is also called as the Nucellar plasmodium. Then there is perisperm. In the absence of endosperm, the ovule encloses cells of the nucellus, and these cells of the nucellus will provide nutrition to the developing embryo. When the cells of the nucellus, they provide nutrition to the developing embryo, this structure is called as the perisperm, which you can see here. It is seen in black pepper. The nucellus does not degenerate as happens in most of the cases. Here in black pepper, the nucellus remains persistent after fertilization, and it is carried on in the seeds, and this nucellus which is persistent is called as the perisperm, and the function of this perisperm is to provide nourishment to the developing embryo, and this is in the absence of endosperm. Then another important topic is genomic imprinting. Now, genomic imprinting is a parent specific gene expression mechanism where certain genes are expressed depending on whether they are inherited from the maternal or the paternal parent, right. This selective gene expression is regulated through epigenetic modifications. Now, these modifications are done by DNA methylation, or there are histone modifications. They will either silence or they will activate specific alleles. Now, this is the whole concept of genomic imprinting. The endosperm, which we have been talking about, is a tissue which exhibits imprinting. Typically, maternally inherited alleles, they tend to promote nutrient provisioning to the embryo, while paternally inherited alleles, they may restrict it, balancing resource allocation. So, this imprinting ensures optimal growth and development of the seed. Disruption in imprinting can result in abnormal endosperm development, which will affect seed viability and will also affect the fitness of plants. Now, this genomic imprinting is implemented to allocate limited resources to the embryo over which both paternal and maternal parents are competing. In angiosperms, endosperm is a triploid tissue. So, this triploid tissue has two identical copies of the maternal genome. We have already talked about this when we were talking about the triploid structure formed by two polar nuclei and one male gamete. So, the endosperm is a triploid tissue with two identical copies of maternal genome and one copy of the paternal genome. Unlike other cells or tissues which are present in a plant, the ratio of maternal to paternal genomes in endosperm is 2 maternal to 1 paternal, which if expressed can cause genomic imbalance, and it can influence the development of the endosperm and the seed, because this is not a balance. Right. There are 2 identical copies of maternal genome, whereas there is only 1 copy of paternal genome. An excess dose of maternal genome can lead to precocious cellularization, and this can lead to a reduced endosperm growth. To avoid this balance, the flowering plants, they undergo genomic imprinting in the endosperm, where male genes are silenced and the maternal alleles, they remain active, resulting in monoparental imprinted expression. Right? I hope you have understood the concept of this genomic imprinting, where some genes are silenced. DNA methylation and histone modifications are key epigenetic mechanisms. These are the mechanisms which are involved in gene silencing. What does it mean? It means they control whether specific genes are turned off without changing the DNA sequence itself, or they are turned on. So, basically what is happening is that in genomic imprinting, especially which is observed in the endosperm, certain genes are turned off so that their expression is not mixing with the expression of the other genes. So, that is what is genomic imprinting, which is seen over here. Okay. So, I would just like to quickly revise that endosperm is a structure which is providing nourishment to the growing embryo, and these there are 3 types of endosperm which we have talked about. One is the nuclear endosperm, second is the cellular, the third is the Helobial. In nuclear endosperm, there is the division of nuclei, mitotic divisions they occur, but none of these divisions are followed by wall formation. At a later stage of development, that is towards maturity, there is cellularization or wall formation which takes place, which begins from the periphery and it moves towards the center, that is, it is centripetal. In cellular endosperm, as and when the nuclei divide, each nuclear division is followed by cell wall formation. The third type of endosperm is the Helobial, which is a combination of both nuclear as well as cellular endosperm. That is, first division, the first nuclear division is followed by wall formation, separating the central cell into 2 chambers. One micropylar is larger, the smaller callosal one, and they now will exhibit cell wall formation partially, and there can be a cenocytic stage also which is present. The primary function of the endosperm is to support the growth and development of the embryo and also lead to seed germination, right. Those seeds which consume the endosperm, those are the non-endospermous seeds, and where the endosperm remains till the maturity, those are the endospermous seeds. Okay. Now, DNA methylation, which we were talking about, it involves adding a methyl group to cystosine bases in the DNA, usually at the cystosine phosphate guanine sites. When a gene's promoter region is highly methylated, transcription factors cannot bind effectively and the gene is silenced. DNA wraps around histone proteins to influence chromatin structure and gene expression, right. And this is all about the endosperm. Thank you very much. We will meet in the next class again.